Okay, in this video, we're going to talk about this word called a blank. 3.5 and 3.6 are titled laboratory reagent blanks and field reagent blanks. Okay, well, what does that mean? All right, well, we kind of already referred to this at some point in time. A blank just means a solution that does not have the analyte that you're trying to look for. So in our previous examples with like the cereal, then this is going to be a solution that really doesn't have iron in the mix. Okay, this is something that is typically the solvent, and it's just the solvent only. Okay, so we've talked about this term matrix, and now we're talking about this term called a blank. And I said, well, the matrix could just be the solvent. And now I'm saying, well, the blank is just really the solvent. And it's like, well, what's the difference between the two? Well, keep in mind that when we had the discussion with the matrix was also a piece of the sample that we were not trying to analyze or actually see for any point in reason. So the cereal sample was crushed, the cereal sample was dissolved. There's things other than maybe the iron content in the cereal sample, and all of that becomes part of the matrix. So it is the solvent plus the other stuff that we're not trying to measure. That's the matrix. For the blank, this is straight up typically just the solvent only. So we are not really doing anything with samples necessarily here. This is just the solvents. That could just be water from our tap sprayer. It could be a weakened solution of an acid, a weakened solution of a base. It could be some type of reagent that we're adding to every single thing, but it is typically referred to as the solvent of my samples and of my standards. All right. So that is in particular to the blank. It should not have anything that you're looking for. So therefore, the signals from the blank should be close to zero because it is blank. There's nothing in it at all, right? So if there's nothing in it, then I shouldn't get a signal from these things when I run them on instrumentation or use some type of equipment in a laboratory when I generate these signals and these responses that these things are going to give me. So there's two different types of blanks that we can refer to in a laboratory. And the first one is called a lab reagent blank. And I'll explain that in just a second. And then number two is something that's called a field reagent blank. So both of these things are a thing. Both of these things we want processed. Both of these things we want kind of in the mix of everything. And we do have abbreviations for each one of these as well. So if I go back to the actual method document, I'm going to see 3.5 lab reagent blank, LRB. That is the abbreviation that we like to use for this. All right, so with the LRB, what is it? A aliquot. An aliquot is just a fancy term that means portion. So an aliquot of reagent water or other blank matrix. That means maybe other stuff is there that's just more than water. Maybe it's more than just acid. Maybe it's more than just base. Maybe it's water plus acid plus something else that got mixed into everything, samples and standards and all. That's the matrix. That is treated exactly as a sample. It includes exposure to all the glassware, the equipment, the solvents, the reagents, the internal standards, and the surrogates that are used with other samples. So this is an idea that I'm going to take a blank and I'm going to treat it as a sample. I'm going to pretend like this is something that has been given to the lab. I'm going to pretend like I treat this like all the other samples that we would in the lab. I'm going to process them the same way, react the same things with them, heat them up if that's a requirement, cool them down if that's a requirement, filter them if that's a requirement. They go through the stages of everything, just like a sample would. But I know there's nothing that should be there. I know that in the end, my signal should be 0 or close to 0 as much as possible because our analyte of interest is not present. Hence, that's why it's called a blank. But a lab reagent blank really should follow the steps of the processing of everything, just like all of your samples do. All right. The LRB is used to determine if the method analytes or other interferences are present in the lab environment, in the reagents, or in the apparatus. So with the blank, notice what it says. Exactly the same, including exposure to all glassware, blah, 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 plus internal standards. So we still add an internal standard to the blank, and surrogates. So we still add the surrogates to the blank as well. So with that said, blanks give me no signal on the analyte that I'm trying to find, but it will provide a signal with internal standards if I add them, and it will provide a signal with the surrogates if I add those into the blank as well. And 3.5 says I should be, right? It says take this reagent blank, take this lab reagent blank, LRB, and you're going to process this like all the other samples, including add the internal standard to them. Therefore, you should see a signal, right? That checks the equipment, making sure the blank was ejected in the right amount when you were not there, and watch the machine do its job. And the surrogate, meaning you added that surrogate into the blank, you know that that surrogate should give you a signal, you know what that signal should be, you know what that concentration should be, so it helps you make sure that that process is taking place accordingly. So that is the laboratory reagent blank, or LRB. So a blank typically is always ran at least with every batch of samples. So yep, that is something that we include. We know going into it there should be no signal with my analyte. We know that it should be 0. We know that there's no math that's going to go on that part at least. But this is again another piece of quality that's getting intervened into the SOP, making sure that our lab data has integrity, making sure that we can put some oomph behind our lab reports when we begin to generate them from a laboratory. Next up are field reagent blanks. You can probably imagine what that abbreviation is going to be. So 3.6 is a field reagent blank. So FRB is what that one's going to be. Just like before with lab duplicate and field duplicate, these are both blanks that are taking place in different areas of the processing of a sample. So if we take a look at this definition for FRB, a field reagent blank, it says an aliquot of reagent water or blank matrix that's placed in a sample container in the laboratory and treated as a sample in all respects, including shipment to the sampling site, so putting it into a van, driving it out to Greenfield Lake, exposure to the sampling site conditions. That bottle is going to be with the person in the field collector the entire time, right? Is it really cold outside? Is it really hot outside? Did they accidentally forget and left this thing in the van overnight? I mean, what happened in the transportation, collection, and preservation? That's what we're testing. So the storage of it, the preservation of it, and all analytical procedures. And the purpose of the FRB is to determine if the analytes or other interferences are present in the field environment. So that's really what we're testing here. That's not surprising. That's why we call it field reagent blank. So this is a blank, reagent water, acid, base, a mixture of 2 or 3 things. It doesn't really matter, but this is going to go into a container, and that container is going to travel with the field collector. And that is going to be exposed in all steps of the collection process, the preservation process, the storage process, keeping it cold, keeping it hot, doing whatever they need to do to those samples when they gather them out there in the field, and then they bring them back to the lab. That entire process is getting checked with a field reagent blank, because what we want to see is that at the end of this, the field reagent blank should still be a blank, right? So therefore, this is going to be treated as a sample, just like all the other things, and there should still be no signal that comes from this blank in the collection process. If there is, then there is a sign that the collection, the preservation, the storage, the transportation piece of this entire method is getting contaminated in some form or fashion by just environmental exposure. And that's something that we can check. It's something that we can just make sure that's not going on. And the only way that we can do that is through a field reagent blank, or FRB. All right. So these are blanks at the heart. Again, no signals. We know nothing's there. We put it in there. It should just be our solvents. Most of the time, that's all that it is. And those solvents are either going to travel with the field collector and be brought back, or they're going to start within the lab and stay within the lab, so that way we can make sure that our lab is not contaminating samples as well. So those are the two blanks that we're going to be talking about that you'll see show up on the assignments, or maybe on a midterm or some type of test: LRBs and FRBs. So lab reagent blanks are going to be tested to make sure that there's no contamination, particularly within the lab. And field reagent blanks are going to be tested to make sure there's no contamination due to the storage, the transportation, the preservation, etc. of those samples when they are collected by a person out there in the field. All right. So LRBs are typically present with every single thing that we run. FRBs are going to be present with every single thing that we run. Again, though, keep in mind that this is in terms of EPA methods. That's really where we were working. And maybe some methods don't require field reagent blanks. I mean, they might not, but we are EPA method specific right now. But these terms do apply the same way to all organizations pretty much across the board if they are going to utilize those particular blanks in their batches and their runs. And a lot of times they will. All right. So that's the story with a blank. Again, instrumentation should read 0. It doesn't have anything in it that I'm looking for. It's an expected value. It's an expected number. I hope that it is close to 0 when I'm running them. If not, where's the contamination coming from? Where is that signal coming from? There's a reason that maybe I'm getting a positive signal, and we've got to find out where it is, so that way I can correct for that or expect that to show up also in my samples when they get analyzed at the same time. Okay, so with that said, that's where this video is actually going to stop. We've got more blanks that we can talk about. That will happen just a little bit later. I'm not going to talk about them right now, though. This is going to give you a very short kind of 15-minute video just to organize some of your lecture notes and make sure that you understand the difference between these blanks, because actually there's more blanks that we need to talk about as well. And I don't want to confuse or put too much hodgepodginess into our lecture descriptions and what these definitions are. So I'm just going to do a very quick break here. LRBs and FRBs are what we'll end up with. In the next video, we'll talk about some other solutions that we will expect to find in a quality control environment, and we'll talk about the purpose and the need for them, and that's where we'll pick up. So take a short break, go have a quick little nap, especially if you've watched me back to back, and come back and pick up from there. So until next time, I'll see you again, and I'll have more definitions ready. But, you know, hey, listen, if you're frisky and you want to keep going, trucking on through and taking some lecture notes and listen to me gabble about definitions again, then come on right back. I'll be ready for you. I'm ready for you now. But right now I'm going to put you on pause. So until next time, see you later.